All right. Hello, everybody. This is me again, Matt, one of the SI leaders for biochemistry this summer. So I do want to, first of all, say congratulations. You all are on the home stretch to being able to be done with this course so far. If you're at this point in the game, that means you've already got through exams 1 through 3, and now you're getting through to exam 4, which is a whole bunch of DNA, genetics, and everything along that line. For some of you, I know it's been such a breath of fresh air because you're not doing regulation with Dr. Xiley anymore and no more crazy cycles and everything. But what we're going to talk about today actually is something that might have got you a little bit unsettled a bit because we start to see another process that you kind of have to learn, and it has some really freaky structures, kind of like what we're looking at here on the screen. And we're actually going to be talking about exactly how does the RNA do the splicing. Because we know that once an RNA transcript is made, there has to be some quick modifications that happen, such as making a 5' cap and a 3' poly A tail. But then there's this whole thing about splicing out your introns and then retaining your exons, and then it starts to get really complicated. And in this course, one of the big tasks they ask you to do for exam 4 is to figure out exactly, or to learn exactly rather, how does the spliceosome take care of basically taking out an entire stretch of mRNA and then joining it back together. So there's a lot of little proteins involved, which are units of the spliceosome, and we're just going to kind of go over exactly how would be a good way to kind of look at it. So I know that for sure we are looking at these parts of the slides, but I actually made some of my own material to kind of go over this. So this is all in reference to this kind of stuff. So we're going to go over to what I made. I decided to draw this all out on paper. I'm a little bit off-centered, my bad. So I decided to draw this all out on paper so that you don't have as much of the other parts of the slide getting in the way, so that you don't have to worry about all the extra stuff. We're just going to focus on what we want to do. Okay, so we're just going to jump right into it. So the whole concept here is that we have some mRNA transcript that is represented by this really long line that extends all the way out to the right and to the left for a good amount of distance, but we're only going to be looking at what happens at a certain intron that we have to get rid of. So we're going to go here with step 1, and we're going to have to first recruit a couple of proteins to the site that we want to splice. So I have kind of done things in color-coded again. Each component of the spliceosome that we talk about is going to have a different color. So we're going to start here. We say that U1 is recruited to the 5' GU site. So this is going to be represented here in purple. There is a GU within this mRNA transcript, and I've kind of drawn it with like a hollow center. U1 is some type of globular protein, so normally it would cover it and you wouldn't see this kind of stretch of the mRNA that goes through, but I kind of left it hollow just so that you can see it, and I know the slides did that as well. So anyway, U1 of the spliceosome is recruited to the 5' GU site, and U2 is recruited to what's called the branch point A. The branch point A is going to be between GU and AG. This is a really important, these two groups right here, the GU and the AG, they should ring some bells because you had to learn, probably right before this in the lecture, what was called the GU-AG rule. And the GU-AG rule means that the introns that you are trying to splice out are marked, or like they happen exactly between a certain GU base and another AG base. So notice I have this highlighted in pink down here. What we're going to be splicing out, and I'm going to carry this color coding through the rest of the video, what we're going to be splicing out is this little part of the mRNA that is in green. Now granted, you see the rest of the line. Those are all A, U, C, and Gs, but the only important ones we care about are the GU site, which is the 5', like if this is the 5' end over here, and then this is the 3', ooh, that's kind of ugly. That's the 3' end over here. The only parts of the mRNA transcript that signal the splicing boundaries are the GU site and the AG. But this branch point A is going to be really important as well, because this is how we're going to be able to pinch off this entire segment in pink. So returning to this, U2, which is a unit of the spliceosome, is recruited to branch point A. Now, why do I have all these really weird lines above A? That's because I'm trying to kind of create a visual picture that when U2 binds right here, it actually creates a lot of pressure and tension on this branch point A. I wrote down here that it causes A to branch out. So lecture 36 does also have a really good picture of this. I believe it's just one slide down from here. Yeah, so I'm drawing the lecture slides, and I am going to be drawing all these U1s and U2s, and there's many of them, but we're going to draw them as nice little circles, but they really look like tRNAs. They kind of have that same structure with all the hairpins forming and the arms. We've seen that before, but this slide does show a really good depiction of it. I had the little lines going like this over here to signal pressure going downwards, but what it does is it kind of forces this A out into some open space, and it exposes a 2' hydroxyl on here. Because remember, any RNA structure, it's going to look the same as DNA structure, except for there's going to be 2 hydroxyls on positions 2 and 3 of the molecule, whereas DNA, the 2' position had, through the enzyme ribonucleotide reductase, lost this oxygen right here. But anyway, this is just a quick picture of what it looks like when the pressure of those lines in my diagrams that I had, pushing down, this is what it's actually doing. So as we go through that, I want you to keep that in mind. But anyway, so back to the stuff that I had written, which is probably just a little bit less cluttered. We're going to go down to step 2. So step 1 is just literally recruiting U1 and U2 to both the GU site and the A branch point, respectively. So now we're going to go on to step 2. Step 2 says that we are now going to have to recruit even more units of the spliceosome. We already have U1 and U2 safely bound on here, and now we have to start recruiting U4/U6. We're going to recruit the complex of both of these 2 units of the spliceosome, and we are also going to recruit U5, which is another unit of the spliceosome. So I have put these in color-coded notation down here. So this was what we had at step 1, and now step 2 is adding these 2 that kind of just start to sneak into this mRNA transcript. That used to be just a nice straight line, but now they're going to kind of wedge up underneath it, and they're going to start to create some bending going on. There's not necessarily any chemistry of this bending that has happened, but it's just kind of starting to push on and make a concave little insertion right here. So U4, U6, I did half of this in red and half of this in this green color over here. So, but soon we'll see that these are going to separate and be standalone molecules. So that's why I did the color coding half and half right here. U4 is this red part, U6 is this green part. And then we see U5 being recruited down here in the yellow. So what this is called is the inactive spliceosome. I'm sorry, that kind of got cut out when I was taking photos of these cards that I made. But so whenever you have this here, it's technically everybody has arrived at the party. All the units of the spliceosome that are going to do something have arrived, and they are associated with this transcript all at once in here. But as we know, the spliceosome hasn't done the splicing yet. So this is what we call the inactive spliceosome. All right, so now that that has happened, you start to see U4, U6, and U5 kind of wedge into position, and now the chemistry is going to start to happen. So we're going to now move on to step 3. So here's what we had at the end of step 2 right here. This is kind of just a tighter picture of it. I kind of ran out of some of the space, but we have U1 and U2, still the purple and the orange. and then U4 and U6 with the red and the green, and U5 is in the yellow right here. So what happens in step 3, actually, because remember, this is the inactive spliceosome, we have to make it into the active spliceosome. So what's going to happen is that both U1, which is over here in purple, and U4, which is over here on this red part of the U4-U6 complex, that is going to leave the system. I say like minus, just like math. Like we're going to minus U1 and U4. They were really there just for like the positioning of things. They're not going to do the actual splicing. So those are just going to dissociate from the mRNA transcript, and they're just going to float away. It doesn't matter. They just go all the way out here. We don't really care what happens for them, what happens to them at this point. So what's going to happen is that we are assembling the active spliceosome, and in order to form the active spliceosome, you kind of have to start manipulating the actual mRNA right now. So notice that the GU that was once occupied by U1 unit of the spliceosome, U1, before it leaves, it's going to say, hey, U6 unit, can you come over here and now occupy and bind to the GU site? And U6 is going to say, yeah, sure, no problem. So what ends up happening is that U6 now occupies the space for the GU site. And this mRNA transcript, this is the intron that we're getting ready to splice out. You can see it's getting into formation. What happens is that U6 and U2 now work together to bring branch point A in between both of them. You can see it's almost like it's getting pinched in there. So it's preparing to be spliced. In the meantime, U5 is kind of just chilling out behind them. It's more so there for stabilization. It doesn't do too much of the chemistry. However, enzymes, they have to work together sometimes to be able to achieve their goal while obeying all the laws of chemistry that say, you must be stabilized by something. So U5 is going to be the main site of stabilization. U6 and U2 are the main key players that are going to get ready to do some of the splicing. So it brings branch point A. And remember, branch point A has this free 2' hydroxyl, and we're going to have to do something to it. So you can see this formation, and it's looking, starting to get exciting, like something is going to happen. So we have the active spliceosome right here. So how does the splicing actually happen? We're going to move into step 4. Step 4, so this is what we had at the end of step 3, just right up here. This is the one where I ran out of space, but it's kind of the same thing. U5's chilling out there behind, just stabilizing everything. U6 is at the GU site. U2 still has a link to the branch point A. Notice that, and I'll kind of scroll up to the bigger picture to show this real quick. Notice that when U6 took over the GU site, it's kind of pulling U2 the U2 subunit closer to it. So by default, the branch point A is being pulled in. So I'm not saying over here that AG is bound to U2. I'm just saying that, you tugged this strand right here over to the left. So all of it's going to move to the left. This is not a bond between U2 and AG necessarily. So now going back down to the home of step 4, we are going to have to form what's called the Lariat structure. So what happens is that this is the transesterification step 1, and we're going to show some actual slides from the lecture PowerPoint about this after this. But if you want, right now, you can label this as transesterification step 1. What's going to happen is that the branch point A, remember branch point A has that free 2' hydroxyl. It looks like this over here. I'll just flash over to the lecture slides real quick. Branch point A has this exposed 2' hydroxyl group that is going to be eligible to do some reactive chemistry. So what happens when we form the Lariat formation is that the A branch point, which is normally right up here, it's going to do a transesterification. Now, I say transesterification. This is an OH hydroxyl attacking the phosphate group that is on that is on this U base right here. Because technically all of these have a phospho-sugar backbone, and then they have the bases A, U, C, and G. So the 2' hydroxyl is going to attack the the phosphate of the G base. So when it does that, it actually forms a bond. But it's weird because the bond is originated with a 2' hydroxyl rather than a 3' hydroxyl. Because remember, the 2' hydroxyl is kind of what is exposed out here. It kind of, sneaks out, and then there's an OH right here. So that's what's going to be doing the chemistry. I'll just erase that real quick now. So that's what's going to be doing our chemistry in the formation of the Lariat structure. I know he wanted you to kind of keep an eye on this name. The Lariat structure is not what is formed at the end of step 3, but it is getting close to being the Lariat structure. It kind of has the same shape up here, but, this is the completed Lariat structure right here. So what ends up happening is that you can see that this is a transesterification, by the way, because, this once had the phosphate linked to this this hydroxyl group of the U base, but now branch point A basically stole it from the G that used to be associated right here. So once that Lariat formation is formed, this entire structure, and I kind of like to think of this as like a lasso, like a cowboy, he twirls the circular rope above his head. That's kind of what it looks like. So the Lariat structure, after that has happened, basically I say LG down here, it basically becomes a leaving group, and this part can float away. The Lariat structure in pink, as well as U6, U2, and the green U5, so I have an arrow pointing at it, that's U5 chilling out behind there, that can all float away. And I'll kind of show you what that looks like in the lecture slides. I'll just scroll down. I think it's 2 more slides down. We were explaining everything that's happening in all of these steps, by the way. And this is kind of a big zoom in on the steps that we were talking about. So once the Lariat structure has been formed, it can all float away. What floats away is the U2 structure, the U6 structure, the U5, as well as the Lariat. That is the intron. The U2, U5, and U6 are what are able to carry the intron away because we are splicing that out. We do not want that to be part of our final protein that will eventually be translated. So this just gets the hell out of here. It's gone, and it has been successfully spliced. But now what happens is that, we have a nick in the backbone. We have a nick in the resulting transcript. So kind of to swap back over to what I was making, what happens is there is this junction right here, and this is what we call the splice junction. The splice junction has nothing to do with the Lariat or the U1, 2, 3, 4, 5, and 6. It only has to deal with what is left over after the spliceosome components take away the intron that you're trying to get rid of. So this is referred to as the splice junction, this little gap right here under my cursor. So, we did the first transesterification right here. This was the actual transesterification happening, but where does transesterification step 2 happen? Well, it's actually going to be involved in fixing the splice junction. So instead of writing this out on my own, I think the lecture slide did a really good job at showing the last part of the transesterification because it's necessary to fill in this little splice junction that's left right there. So we have to take a look at exactly how this is happening. So I'm going to go over this slide with you. He really stressed that you guys know the biochemistry of this slide. So if you take a look right here, the words do a really good job at explaining what happens. In the first transesterification reaction, the 2' hydroxyl of the branch point A, because remember that branch point A, it had this 2' hydroxyl due to the pressure of the U2 spliceosome subunit, basically bearing down on it. The 2' hydroxyl of branch point A residue attacks the phosphate at the 5' end of the intron. So this slide explains both transesterification step 1 and 2, but let's take a look at both of them, kind of. So over here to the right, I have an RNA kind of, this is when RNA is linked together. Notice it's single strand. There's not another strand of RNA over here, out here to the right, because RNA usually is single-stranded until it forms, hairpins or stuff like that. But it's not like DNA where it has a double helix in its main state. So the first transesterification says the 2' hydroxyl of one of the bases, which is branch point A, that is a base. It's just like, it could be this one, for example, it is a base A in a chain of RNA. So it would look like this guy right here in the yellow, and then, including its, sugar backbones. But if we take a look at the numbering of, and you guys should probably know this, but we'll review it right now real quick. If we take a look at the numbering of the carbons of the sugar backbone of the RNA molecules, because, the RNA includes the bases A, C, G, A, and U. It includes the bases, but never forget about the phosphosugar backbone. So carbon 1 is going to be at this vertex where my cursor is. Carbon 2 is going to be at the bottom right. Carbon 3 is going to be at the bottom left. Carbon 4 is going to be at this vertex on the left side. And carbon 5, don't forget about this guy out here. There is a kink right here before you get to the phosphate. This is carbon 5 out here. So when you say that the 5' phosphate, it is the carbon that links to these little kinks out here, because this is the 5th carbon. And then 4, 3, 2, 1 over here. And, the glycosidic bond is formed between the base and carbon 1 of the sugar backbone. So we say that the hydroxyl of the 2' hydroxyl of branch point A is going to attack the 5' hydroxyl of, basically the G of the GUAG rule. Because you have GU over here, and then you have branch point, if this were like a transcript, you would have branch point A right here, and then you would have GUA, and then, well, no, there's branch point A, and then there's an AG out here as well. But, so just, if we were to line this up, that's kind of where it would be. But I'll erase that now, because I just want to draw that as a quick, just kind of example. But so the 2' hydroxyl, so I know this one isn't the A is technically down here, but I did the numbering up here. So pretend this is an A up here. Pretend that's an A. The 2' hydroxyl, it's saying that this hydroxyl group is going to attack carbon 5. So it would be this one at this vertex. This hydroxyl is going to attack carbon 5. And it's basically going to steal the phosphate group from the G base, because this is exon 1. You retain your exons and you splice your introns. This entire loop up here is the intron, and one of those bases in there is branch point A. So do keep in mind that this is the G basically right here, and the 2' hydroxyl attacks the phosphodiester, or the phosphate group, and then it forms a new phosphodiester bond to make the lariat. Notice that this is, it steals G along with it, but what is left over is the 3' hydroxyl of, the base that was next to the G and the U. Like if there's G and then there's U, there's another base, I'm just going to call it X out here, that was right here. So this is, X isn't a base, but, there is some base out here that now has a naked 3' hydroxyl. And that is what this is what forms the splice junction. The naked hydroxyl, and later on, when you take out, so to switch back over here, it's the naked hydroxyl that is left over right here. And then, there's another base X out here that has its own, missing end as well. So that's going to be, this missing end out here is going to correspond to the phosphate. Yeah, because there's a naked hydroxyl and then there's a naked phosphate. You need to make a 5' to 3' bond between the phosphodiester to make the phosphodiester bond. If this is 5' right here, that's 3' hydroxyl when it's naked. If you kick this out, there would be 2 hydroxyls right here because it's RNA. So The second transesterification reaction is what's going to seal this splice junction. Once you form the Lariat structure, the naked 3' hydroxyl is going to be like, hey, I don't like being here alone. I need a phosphate group. So it's going to steal it from the 5' hydroxyl of whatever the next base would have been, because you have, to go back over to my thing again, you have the GU and the AG. Basically, GU, the G and the U, it's kind of backwards now because we kind of swung it up into the air. It links with branch point A, so this hydroxyl is going to attack the hydroxyl of technically whatever base is over here, because what gets sent off in, the entire splicing, this transesterification would not attack the AG that we saw over, I know I'm flipping around a lot, but the second transesterification will not link between this OH and the AG, because that is also included in the intron. That is what gets sent off and it goes away. So to show you right here, just one slide up, you can see that both the GU, and remember it's kind of flopped because it's swinging up in the air like a lasso, the GU and the AG gets released. This exits the system. So whatever base would have been after this AG, so right here, whatever is the next base in this part of the green right here, that is what this OH would attack in the second transesterification reaction. This slide does a good job at showing the intermediate structure and the phosphates that are happening, but it doesn't mention that this is the 3' of not the GU or the AG. This isn't the phosphate of the AG, and this is not the phosphate of the GU. This is some other base, which I called X, that was right before the GU and the AG sites, because the GU and the AG and everything in between that, if there's like a GU and an AG, I'll just draw it over here. The GU and the AG and everything in between it, if that were, I know I'm kind of drawing over things, but everything in here, including these things, because you remember the GU-AG rule, those are the boundaries of the splicing. So X here and X here. When we say that the 3' hydroxyl attacks the 5' phosphate of the second transesterification action, it's this X over here linking to this X over here. So I hope that is clear, and I know I'm kind of drawing all over the place, but I'm sure that you guys can follow that part at least. My drawing. But anyway, that's what's going to resolve the splice junction, and then from there, you don't need like DNA ligase to seal the nick. It's really tempting to say DNA ligase is going to seal that nick, but do keep in mind we're working with RNA over here, and it does its own transesterification reaction to be able to get that to work. So yes, that is basically everything I wanted to go over in this video. I hope that that makes everything in terms of the spliceosome function, the actual splicing of that intron and getting it the hell out of there, because, we don't want that to be in our final protein. Well, depending on alternative splicing, which sometimes you do want to keep it, sometimes you don't. But in general splicing, that is how you get rid of an intron. You have the U1, the U2. Well, I don't think, does U1 exist? Yeah, there's the U1, the U2, the U3, 4, and 5. I actually don't think U3 is in here now that it's just U1, U2, U4, and U6 complex, which then soon separate to be U6 and U5, or U4 and U6. They're going to separate, but at one point they are kind of linked together. So it's 1, 2, 4, 5, and 6 that take care of all this splicing. And the GU and the AG and everything in between is what's going to leave. That is basically the lariat right here. That looks like the lasso being swung above the head. And don't forget, all this time, the branch point A is going to be a key instrumental player in the chemistry because it donates that 2' hydroxyl group that does the first transesterification reaction to basically steal U and G from the 5' end over here. So that's everything I wanted to go over. I hope you guys enjoyed this. And once again, happy studying. You're on the home stretch of this course. And I know I didn't say it at the beginning of this video. I'll try to put a a little notification up here in like the top right or something like that that says you can speed up this video if you want, but that is totally up to you. Perfect. Have a great rest of the day.